This is a production of Cornell University.
Okay, I want to thank Penelope and the students from the Boyce Thompson for inviting me in. And this is the first time I've ever been called a translational taxonomist. And in fact, this is a term, translational taxonomy is a term I coined. And so having that term be actually applied to me is kind of interesting because nobody knows what I'm talking about until I give, a talk about it. And then they're like, oh, that's what it is. I tend to refer to myself as a phytobacteriologist or sometimes a bacterial taxonomist, but a reluctant one at that. But it is a lot of what I do, in fact, or how I approach research. Really want to thank you and your cohort for bringing me up here, and I learned a great deal from you all, and I appreciate that. Just an aside for mentoring, I believe that mentorship goes both ways. It really is a relationship, and that mentors often learn as much from their mentees as the mentees learn from their mentors. And so that's the approach that I've taken. So I've learned a great deal again here. In fact, I learned about aptamers yesterday. And so if you don't know what aptamers are, there are people here that do. And I'm going home to research them because I think they could be really, really powerful. Okay, so we're going to talk about translational taxonomy for bacterial pathogens. Just to, whoops, of course. Hello. Just to give you an outline of what we're going to talk about today. Really want to talk about the complicated state of bacterial taxonomy. Many people think it's complicated. I don't. I can keep lots of information in different systems in my head at one time, and I like to think about it. So it's not complicated to me. So when people say it's complicated, I think, not really. However, I understand why people think it's complicated, so I want to explain that. I want to give you some examples of what I mean by translational taxonomy, and I want to give you a little bit of where I've come from and where I'm going in the lab, what we're doing right now, so you have a feeling for how you might be able to collaborate with us, for example. And then briefly, I just want to say a few things that we're doing at PSU in general. And I want to do that because that's where I spend a lot of my time these days, is as a department head and head of a research center. I spend a lot of time doing other things than the research. So the people who do the research, these are some of the pictures, and you'll see a little bit of their work as we go through. Okay, so as Penelope said, I spent 20 years in the Salinas Valley of California working for the USDA. Some of the research I'm going to talk about was done related to the Salinas Valley, and so, but much of it is translatable to Pennsylvania because almost everything, except for maybe avocados and citrus, that was grown in the Salinas Valley, and that's avocados and citrus aren't grown there, but they're grown in California, is also grown in Pennsylvania. So a lot of the same kinds of questions can be asked in a different context. And in fact, I'm still collaborating across the board on some of these crops. These are the crops that I worked on when I was working in Salinas, and some of the crop values, at least of 2012, the values of these crops. I just note that, for example, if we look at cabbage, I'll just take an example. The cabbage production in Pennsylvania is worth $3 million. Instead of 35 million. And that kind of gives you the flavor for the whole relationship between maybe the Salinas Valley in the salad bowl of the US is what it's called, versus what we're doing in Pennsylvania. But to the growers that are producing these crops, they're extremely valuable. And so thinking about disease systems and how to help those growers with the diseases is important in both places. So what is translational taxonomy? So I think of it just like we think of translational medicine, which is research, basic research that is brought into a clinic. I think of translational taxonomy in the same way. So it's looking at the three pillars or the legs of the stool of taxonomy in order to develop strategies for disease management, which is the seat on which we sit. I'm interested in all three pillars of taxonomy and in relationship to bacterial plant pathogens. And I'll just give you a little bit of what that is like. And I'm going to give a little bit about bacterial taxonomy. My goal is that you're all awake at the end of the bacterial taxonomy part, so that we can get into the disease management part. Okay, so that is my goal. We'll see if it works. Let's see if people are nodding off or not. Because often bacteria taxonomy in general, people start to glaze over, and yeah, it's something that I do when I need to fall asleep at night. Okay, so these are some of my key interests in basic taxonomy, and I just want to put them out there so that you know that I am a little bit nerdy. In that I really do find pleasure in reading about species concepts and thinking about species concepts for bacteria. In particular, in plant pathogens, I'm going to explain a little bit about the Pathovar concept and how we use the Pathovar concept and whether or not it fits in some systems. And I'm really interested in where it's breaking down and why it's breaking down. And I'm also interested in this question of natural host range versus experimental host ranges, and what that means for our classification systems. And so I'll just put that out there. I personally want to get rid of the binomial system of nomenclature, and I'll just say that. If you want to talk about that later, it's another hour seminar. But I would like to junk Linnaeus. I think he did a great job of taking the paragraph that was the name of organisms down to 2 words. That was really helpful, but I think now we need to take it to 1. And then I often talk about identification in with regard to the appropriate level of taxonomy. What is your target, and what do you need to at what level do you need to identify something to actually control the pathogen? And often researchers, we want to get to a more refined level than a grower might need. Or the grower doesn't know that they need to be at a more refined level than they are. So having the appropriate taxonomic level understood for the management practice is one of the places where I go to when I'm talking about translational taxonomy. Okay, so just a background about taxonomy in general. If you think about bacterial taxonomy, originally the taxonomy was based on a very few principles or very few characteristics. And it was really based on those characteristics that we needed to distinguish those things that would kill us from those things that won't. Okay, or those things that would kill our plants versus those things that won't. So initially, the basis of all taxonomy for bacteria was really based on a few principles, and they were specific. And you see here listed a few of them, and what the result of that is that the groups that were classified were not really groups that potentially were evolutionarily coherent, but they were groups that looked alike and smelled alike, let's say. And what that resulted in were species that were monothetic, that means they had similar characters, and were polyphyletic, which means they had different evolutionary backgrounds. This, to us now, seems very unusual because we're used to trying to put groups into monophyletic groups and look at their evolutionary history. But if you think about it from a historical standpoint, that's not where we started. And this is really the basis of where a lot of our confusion comes from. And I'll just, we're going to talk about Pseudomonas a lot, and the folks who took the workshop heard my spiel about Pseudomonas yesterday. But pseudo, fake, monas, it's the Greek word for unit. The word money comes from the same word. Okay, so it's a fake unit. They put a lot of things in there that looked alike, but they knew it wasn't really a coherent group. And we've been paying for that ever since, okay? Okay. On top of this subjective era of classification where we needed it for our health and well-being, put on top of that this idea of the new host, new species cliche that has been written about. Again, few comparative tests. When I isolated something from broccoli, for example, I might not have had at the time the ability to compare that pathogen to pathogens from all over the world or from other hosts, in fact. And so there were few tests that were done comparatively. And there were lots of different organisms that were isolated that were actually synonyms for the same species. But with each new host, people were giving them names and designating them as species as if there was a direct relationship between the host and the specificity of that species. And that isn't necessarily the case, or wasn't necessarily the case, but there was really no way to compare broadly these organisms. This is a second factor that has really added to the confusion in Pseudomonas taxonomy, okay? So this led to a number of misconceptions and a lot of synonyms within the literature. Everybody with me so far? Okay. Nobody's sleeping yet, I like that. Okay, so layer on top of that the advance of our technologies, right? So we went from phenotypic testing and maybe host range testing into looking at characteristics that are actually more amenable to hypothesis testing. So if we talk about nucleic acid-based techniques for classification, you can actually start to do statistics. You can do that even with phenotypic testing. But we begin to think about using numerical taxonomy or statistical methods for analyzing these data. And now we're at the point where we can actually look at whole genomes as well to see whether these organisms are clonal, are they the same, are they different, and then how different is, of course, the million-dollar question. But as we layer these pieces on top of that background of confusion, if you will, or of subjectiveness, it becomes difficult, and it becomes difficult to tease apart what is the subjective background and what is the objective data. And I'm going to just show you a little bit of that as we go forward. So at the same time as we were moving from the subjective era of classification, and I could spend a lot of time talking about that era in particular, but as we were moving from that era into a more objective era, there was also going on in the bacterial community reorganization of the code of nomenclature. So remember, classification is one part of the stool, but once you've got the groups classified, then you have to name them. And naming is actually done by a set of rules. Classification is done using the scientific method, so there's question, inquiry, there's data that support hypothesis, but actually naming the organism has a set of rules. There are no rules for classification per se, or there shouldn't be, but for nomenclature there are. And the nomenclatural codes are where the rules are. People often confuse that. They think that the classification has some kind of rules and that there's an appropriate classification. There are best practices according to the scientific method and the thoughts and opinions of the scientific community, but they're not rules. The rules are here in the nomenclature. Okay, so the bacteriologists were reorganizing the nomenclature. In fact, they knew that there were synonyms throughout the bacterial nomenclature and throughout the species that were described. And this is not just for plant pathogens, it was also for human pathogens as well. So they got together and developed a new code of nomenclature. And part of the new code of nomenclature required that there be a catalog of names for valid names of bacteria. And that, getting into that catalog required that a name had a validly published epithet, and that means it was published so we could all read it. It had a modern description, so something that we would all be able to test and distinguish one organism from another. And then there had to be a nomenclatural type strain that was deposited somewhere that we could all get a hold of it and compare our strains to. The problem with this is that it didn't, for plant pathologists, the problem is, it didn't include host range as part of that modern description. So most of our species, in fact, were based on this new host, new species cliche. And all of a sudden, most of our species were going to go away and disappear, in fact, be consolidated. And this is the result of that approved list in 1980. So for example, the Pseudomonas syringae, there were 41 nomen species, and most of those were consolidated into Pseudomonas syringae. Okay, and the problem with that is that we still needed to be able to distinguish the pathogen from celery from the pathogen from tomato. And we could distinguish them based on host range, but not at the level of the species and that modern description. Caused a lot of confusion. So what plant pathologists did, and I know some of you all know this, is that we took the nomen species names, and these are the specific epithets for all of these species, and except for those that are in bold, they were all put into Pseudomonas syringae as pathovars of Pseudomonas syringae. Okay, those in bold were all left as species because they could be distinguished by modern methods. Okay, so the Pathovar concept was put together as part of the International Society of Plant Pathology's Committee on the Taxonomy of Plant Pathogenic Bacteria. Say that 10 times fast. And they developed this idea of a pathovar, which was supposed to be temporary. So the idea is, okay, we'll put them all in Pseudomonas syringae, for example, and as we have all of this money for bacterial taxonomy coming through our governments, we'll be able to tease them apart into real species again. Okay, you know there's no money for taxonomy, and so over the 40 years that since this has happened, is it 40 years? 80? Almost 40 years, you know, we've had some progress, but it's been piecemeal. And that is really the problem with it, is that we've had a piece of work done here, a piece of work done there, and then trying to understand what that whole piece means is what's difficult. Yeah, we'll go on from there. Okay, so this concept of pathovar, really a pathovar is designated as something that has a different host range or causes a different disease, but is within the same species, okay? Now, we don't know whether or not, we didn't at this time know whether or not these things were actually the same species or different species, and this has caused a lot of confusion. Originally, there were pathovars for the 4 species that are listed here. Currently, we have pathovars for over 26 species, just to give you an idea. Okay. So for plant pathogenic bacteria, now you don't just have issues with naming bacteria that at the species level, which are coded by the International Code of Nomenclature for Prokaryotes. Seriously, if you want to sleep, that's the thing to read. But we also have a code of nomenclature for naming pathovars. And you have to work with both of them if you're working with nomenclature of plant pathogenic bacteria. It's actually the kind of thing I like to do, so I belong on the boards of both of those groups that do that. And the issue is that we've got species, subspecies, pathovars, but often we're really working at the level of ecotypes or strains even when we're talking about trying to control diseases. There can be differences between one ecotype or serotype, or it could be any different kinds of characteristic that you've been evaluating. There can be differences between strains at very fine levels, and that may be the level of taxonomy that we need to be working at. And the pathovar may help, but it may be at this lower level that you want to pay attention. Okay. So just a little more confusion for you. So we've got this group of organisms that have been put together, and now we have a concept for species that was layered on top of that. So also around the 1980s, there became this idea that there would be the golden rule for bacterial species concepts, if you will. And this became a species definition. And basically, people found that if they did DNA-DNA hybridization and the DNA hybridization values were 70% or above, that they were likely the same species. And just to tell you what this process is, because it's complicated and not many people used it, you take 2 different bacteria, you label one of the DNA from one of the bacteria, usually with a nucleotide of some sort, and that is labeled, and you melt them, mix them together, and then you let them hybridize, use S1 nuclease to cut away the single-stranded portions. You're left with double-stranded DNA, and then you can measure the radioactivity in your sample and compare it to a sample where you've put the same organism DNA with the same probe. Right? So you've got the organism with itself, the organism with something else, and the difference between those is the percent hybridization. So for any experiment where people wanted to look, is this in that species or not, they would have to use all of the controls, necessary and do each of these hybridizations, because you couldn't create a library of them and just go online and look. So each control had to be done. That means that everybody had to have those type strains or pathotype strains, and people just didn't do it. In fact, it resulted in only a few laboratories being able to do taxonomy at the level that we needed to have taxonomy done. So for a period of about 10 years, only a few people in white towers, very esoteric, who only wanted to speak to themselves. I'm kidding. Don't, wait, this is being broadcast. There was a, let's see, it was not democratized, okay? And so it was very difficult for having normal plant pathologists to be able to determine the species of an organism. Okay, fast forward. And one of the problems with this is that if a species was determined, let's say in the case of Pseudomonas syringae, for example, it was determined that all of those pathovars actually represented 9 different species. But they couldn't distinguish those species one from another using phenotypic characteristics. And that's a problem. If I can't do the DNA-DNA hybridization in my lab, I need those phenotypic characteristics in order to be able to look at whether or not organisms that I'm isolating really are one species versus another. So the nomenclature would not allow us to name species that were really, these are species. The Pseudomonas species is a genetic species that we understand to be related to one another, but we weren't allowed to name them because of the code of nomenclature, because there weren't characteristics that would distinguish them in our laboratories. So now we've got a system where we know that there are 9 species, but we can't actually name those species. And so Pseudomonas syringae still had all of these species. It had 9 species in it, and most people had no idea which was which. Okay, so we know that science is iterative, and classification is an iterative process. So when we start out with the phenotypes and kind of the mess that we had in the beginning, and then we start to tease these things out using the DNA hybridization, that goes on top of that. And now we've got sequence data that we're trying to add on top of that, but we haven't really even cleaned up what we know about the genotypes yet. And so there's a lot of confusion. And where I think we're going for species concepts, we're going to get away from sequences pretty quickly, and it's going to be function as well as interactions in conjunction with the sequences that are going to be the basis of the forthcoming species concepts. Okay, and if anybody wants to talk about that, that's a whole nother seminar too. Okay, so this is the current status of Pseudomonas syringae taxonomy, and I just like to present this to you because this is a clarification of it. Okay, I'm glad you think so. So I just want to show this to you because I think it helps formulate this. So in gray, you have basically a genome species. But people have used sequence analysis to look at the relationship between these organisms, right? And so, for example, Pseudomonas species 3, which would be Pseudomonas syringae or Pseudomonas tomato if it was ever proposed to be that, but the type strain would be Pseudomonas syringae pathovar tomato, DC 3000. Some people know that here. This was designated as phylogroup 1 by people using sequence analysis, okay? And so some people will refer to this group as phylogroup 1, some people will refer to it as Pseudomonas species 3. So Pseudomonas syringae is Pseudomonas syringae sensu stricto, okay, the strictest sense of Pseudomonas syringae, because the type strain for Pseudomonas syringae is in that group. And so here are the different genome species or species. So for example, Pseudomonas cannabina, it's one we're going to talk a lot about, is a species which many people here, some people here work on organisms in this group, okay? But was previously, these were thought to be Pseudomonas syringae, but now people know they're a different species. Okay, everybody with me? Anybody reacting like, no, they're not? Sometimes you do. The only time I've ever seen people almost fight, like duke it out, was about this kind of stuff. You can't change the names of things on people, they get really upset. Okay, so what happens when we get a new pathogen that it might be a Pseudomonas in the lab, right? We test the phenotypic characteristics to begin with when we've isolated it, and then we go on to do the genetic and taxonomic analysis in a little more detail. I'm going to tell you a couple of stories now. How much time do we have left? Tell me a half an hour. Okay, here we go. I'm going to tell you a couple of stories about how we use translational taxonomy now. So between 2002 and 2010, there were a number of outbreaks of disease on parsley. And these were fluorescent pseudomonads according to the phenotypic tests that we traditionally do. Okay. We knew that there are two different pathogens that cause disease on members of the Apiaceae. So there's Pseudomonas syringae Pathovar coriandricola, which causes disease on coriander or cumin, and there is Pseudomonas syringae Pathovar Apiaceae, which causes disease on celery. Okay, and they've never been reported, in fact, on the other crop. So we thought, well, maybe the parsley pathogen was one of these two things. Okay, so the approach that we have traditionally used in my lab is a MultiLocus sequence analysis. Now we're moving into whole genome sequence analysis. We can talk about that later. Anyway, I think you all know what MultiLocus sequence analysis is. You sequence a number of housekeeping genes, you analyze them as if they were one gene, and that gives you a stronger phylogenetic signal. And in fact, we were able to show that these are, and you really can't see this in the translation. So these are different genomos species of Pseudomonas syringae. So these are the different species we talked about in that chart. And you can, you actually, with the MultiLocus sequence analysis, there's really a good correlation between the MLSA information and the genomos species or the DNA hybridization values. So using MultiLocus sequence analysis, you don't need to use DNA hybridization. And in fact, if you look in the literature, almost all of the plant pathology literature now will have MultiLocus sequence analysis instead of, or in addition to DNA hybridization for taxonomy. Okay, and it's a really good system depending upon which genes you use. So if we analyze this a little more closely, the parsley strains came from both Genomos species 9, or Pseudomonas cannabina, or from Genomos species 3, which is the tomato group. Okay, so they were closely related to Pseudomonas syringae Pathovar Apiaceae, but in fact, the API, if you look at the, I do have a pointer, don't I? I just like to move. Okay, I didn't get my workout this morning, so if you look at, okay, if you look at the type strain for Pseudomonas syringae Pathovar Apiaceae, it's quite a bit different than those BS strains, which is bull salinus, as opposed to other BS. Quite a bit different than those other BS strains up there. And so there's been a change in what we're isolating, both from celery and from now parsley. So parsley strains, Pseudomonas syringae Pathovar Apiaceae, but they're also Pseudomonas syringae Pathovar coriandricola in the Pseudomonas cannabina group. So here's a place where we actually have two different organisms causing the same disease on parsley. Okay, and I can tell you that you can make the Pseudomonas syringae Pathovar coriandricola, you can make that pathogen a pathogen on celery, you can inoculate it and you can get some kind of plant reaction, but we've never seen it from the field on celery. And likewise, we can get a plant reaction from Pseudomonas syringae Pathovar Apiaceae on coriander or cilantro, but in fact, we never see it from the field. Okay, so there's a difference between what we're seeing in the fields and what is actually causing disease and what we isolate or what we can get to cause disease or plant reactions in the greenhouse. So what does this mean to a grower? So in the Salinas Valley, where the land is rented for $2,000 to $3,000 an acre per year, it's really expensive land, that's just the rent. They turn over crops really quickly. And in fact, if somebody is a celery grower, they might grow some of these other crops, but they're not going to be growing lettuce, or they may not grow some other kinds of crops. They usually stick within the same plant family. So if somebody's growing parsley and they have the disease, we really recommend they rotate out of the Apiaceae completely. But if they're growing celery and they get the disease, they could go and plant cilantro. So there's a crop rotation system that they can use that they won't see the pathogen again. That's one option, okay? So this is the kind of strategy we use once we know what the organisms are and what the host ranges are and what we're seeing from the field. This is what I consider translational taxonomy, okay? Is everybody with me? Some people are going, oh, maybe that'll work. Okay. I want to tell you another story. This is the story of pepper spot of crucifers, and I want to tell that primarily because I know people are working on Pseudomonas syringae, Pathovar, maculicola, and organisms like that here. So this is the pepper spot. This is some symptoms of pepper spot on this. I'll just tell you that McCulloch was a plant pathologist at the USDA at the same time that Irwin F. Smith was the leader of the program there. And you notice that that publication is McCulloch 1911. At that time, women were allowed to publish without their supervisors on the publications. So Irwin F. Smith, who was considered the father of phytobacteriology, had a number of women that worked for him that published independently and did independent research. And I think it's an interesting historical fact that happened during the 1920s or early 1900s. And then like 30 years later, during the 50s, I guess that's 40 years later, I can't count. Anyway, you know, later women were not allowed to publish. And in fact, often women did a lot of the work and were not even included on the publications. And so I think that's a very interesting historical fact. Nonetheless, she isolated Pseudomonas maculicola. It was called something else when she isolated it. And many, many people work with Pseudomonas syringae, Pathovar, maculicola, but in fact, they're working with Pseudomonas cannabina, Pathovar alicelensis. This is an organism that has been studied for a number of years, and I just want to show you a little bit about how we determined that this really was a cannabina. The host range for this organism is different than the Pseudomonas syringae, Pathovar, maculicola host range, in that it is a pathogen of monocots. And there are a number of crops that we'll see the pathogen on that we don't see Pseudomonas syringae, Pathovar, maculicola on. When we first saw this organism causing blight on arugula or on broccoli raw, we thought it was Pseudomonas syringae, Pathovar, maculicola because it was coming off of a crucifer and it was a Pseudomonas syringae. Okay, so one of the organisms that's worked with, you know, as a model for Pseudomonas syringae, Pathovar, maculicola is ES4326. And I think people here work on it, right? Anybody ever see that number before? It's a great organism. It causes devastating disease on radish. And I know, it's a great organism. It kills things. Okay, the thing is that there have been comparisons between this organism to Pseudomonas syringae, Pathovar, tomato, and there have been explanations of, you know, molecular explanations by why they're different pathovars or why different, maybe different races or things like this, but they're actually different species. And so our thoughts about what this organism is doing and what the molecules that we're analyzing are doing have to be broadened to realize we're looking at two different species. Okay, and I know that's kind of some days shocking. So again, this was the different Genomos species. Here's Genomos species 9. So this is ES4326 in an MLSA, and it is identical to the pathotype. There's a paper where we actually look at the whole genome sequences of these organisms, and there are some differences when we look at rep PCR and the host range. So when we isolate from arugula and radish, we only see the ES4326 type, which is this organism here. Now, other plants we can isolate and we'll see Pseudomonas syringae, Pathovar alicelensis, the pathotype type. So there's an interesting difference there that, yes, you can inoculate those plants and get disease, but really in the field what we're seeing is only one type or the other depending upon the host range, okay? But both of these go to both crucifers and monocoques, and that is a really exciting place that people are working as well. I should say that you can see there's Pathovar Philadelphii and then the coriander here. I have a student, Beth Elger, who is now at Michigan State, who we're working on a paper to go ahead and change the nomenclature of all of those as well. Because we know that you love when we change the names of organisms. Okay. And I'm sorry, I don't know when the seminar ends. So what is the time?
1:05.
1:05. Okay, we're going to forget the cucurbit one. I just get talking. Let me talk a little bit about mushrooms. So we're using the same kind of technique to look at mushroom diseases. And so this is bacterial blotch in mushrooms. There have been attempts to control this disease using bacteriophage, and I've worked with bacteriophage in a number of different systems. And what's interesting about it is that in a mushroom production house, you really have a contained system. So when I think about biological control, that seems like a natural place to be able to deploy biological control. The problem is we know from the literature that there are a number of different organisms. That's how we do pathogenicity testing on mushroom tissue. There are a number of different organisms, keep going, that cause diseases on mushrooms. And so blotch can be caused by Pseudomonas thalassae, which is what is traditionally thought of as the blotch organism. But there's also Pseudomonas gingeri, which is up here, and then a number of other organisms have been shown to cause the disease. So we wanted to know what's causing disease in mushrooms in Pennsylvania. So what we did was survey, we've surveyed so far 7 different mushroom houses, and this shows you the sampling array from 2 beds of mushrooms that we looked at in one house. And for each of these circles, if there's 2 colors, that means on one cap, we isolated from 2 different spots and got 2 different species from that same spot. Each color represents a different species. This one had 3 different species on that cap. So there are a number of species, we think at least 7 species of the pathogens causing blotch on mushrooms. So when someone came, the researchers in the past have looked at using bacteriophage to control this disease. They had a bacteriophage that was effective against 1 organism, but there are more than 1 organism there. And so we're actually working with a class to isolate bacteriophage to all of these different pathogens so that we can try using a cocktail of them and as we monitor what organisms are present in houses, in different houses. And I'll just move forward. We're using a similar approach for blackleg and soft rot of a potato, and I know some of you know that there's a Dickia danthicola is now and has been detected in the northeast and of course across the U.S. And so I have a new student, Amanda Mignello, who is working on this, and she's really interested in looking at a microbiome approach to doing this. Okay. I'm going to skip through that and just tell you that Emma Rosenthal, who was in Greg Martin's lab, is currently working on one of my favorite projects, which is a lettuce disease problem. This is bacterial leaf spot on lettuce that she works on. And we have already characterized both the diversity of the organisms that cause the disease, but we also know that there are 3 races of the organisms that cause the disease. And we have differential hosts for that. We know that at least for one of them, there's a single gene for resistance. So Emma is currently we're working on classifying these organisms because they're not Xanthomonas campestris, they're really Xanthomonas hortorum. There's another name change, sorry. And she's done a great job. She's actually sequenced 27 strains and has assembled those for whole genome analysis to really understand what these differences are and what are the factors that are related to the race relationships. She will be using forward and reverse genetics to do that. So that just gives you an idea of what we're working on now with that particular group. Okay. If I have a few seconds, I just want to say something about the department. This is who we are as a group. We're currently 26 faculty. We run usually around 25 to 26 graduate students, although we've been asked to push that up to 40 by our dean, and so that's an interesting thing. Everybody is going, how many graduate students do I need to have? Yeah, and so that's a kind of interesting goal, and we have some strategies to get there, but that's who we are as a group. We have 4 different signature areas that we concentrate on, and within each signature area, we actually have 4 different areas that we, techniques or strategies for researching these areas, if you will. So whether we're in mushroom science and technology, we use these 3 different or 4 different emphases to look at that area. And so that'll give you kind of a summary of who we are research-wise. We have a new strength in phytobiomes. We've hired 2 people in particular, but we're part of a larger group. And that's what this is. So a year ago, a year ago, we started the Microbiome Research Center. And we currently have 100 scientists who are involved in this. We have 10 that are a part of a cluster hire, 5 that were hired in medicine, 5 that were hired in agriculture, and there are about 5 more that'll be coming on board, 3 in ag. So the College of Ag has made a huge investment in this area. And they asked me if I would lead the center primarily because they wanted to have a really strong foundation for the faculty that they're bringing in to be able to collaborate together, and they're doing a great job. I'd love to talk to anyone about how we've organized this center, but we have a lot of interaction, even with the arts. For example, we're having a symposium in the next, in November, we have 2 different sessions that we'll be having at our weekly meetings that are about arts and microbiomes in ways that I never, like, it's really good I'm not controlling it because I would have never thought to do what they're doing. So we have a documentarian who is very interested in microbiomes, and what that person is doing is very different than a plant pathologist would have a documentarian do, quite frankly. And so that's kind of exciting to me. But across the board, there's a lot of different kinds of interactions, including with our social scientists. I'll just say that we have a lot of different initiatives within the department that comes with having a new department head. The Microbiome Center is key to our strengthening our environmental microbiology emphasis, but we have strengths in STEM and access to STEM and diversity. We're building a couple of bilateral programs with international institutes. The primary one is with the University of Pretoria, and that's been very successful, and we have gotten a lot out of that. The workshop that we had today is part of our comprehensive professionalism training, and one of the things that I've gotten a lot of pleasure in is really engaging with the alumni in the industry. So I think that's it. Oh, last thing is just to advertise the degrees we have. I think 2 things that are unique about our program is we have dual titles with plant pathology. So our students walk out the door with a PhD in plant pathology and maybe a PhD in biogeochemistry, or plant pathology and international ag and development. We also have an online certificate in ag, biosecurity, and food defense. And we have a couple of minors. We don't have a major in plant pathology for the undergraduates, but we have minors, which feed our programs, feed your programs, in fact, and feed other people's programs. And so more the merrier. And there we go, my acknowledgements. And I want to thank you all for your patience and listening to some of the things we do in my lab. Okay, any questions? That's my current team. That's the current team.
Not sure who's on Zoom, but you might ask who's on Zoom.
Who's on Zoom? Who's on first? I don't hear anything. Okay. And I've run right up to the limit. That's what happens when you, so I have to say, I planned this talk last night. I decided that I wanted to do something different than I've done before. And so this is my first time trying to talk about these things in this way. And so I'll try it again next time. Maybe it'll be shorter.
I was curious, you gave a really elegant example of the deciphering some of the entities associated with these diseases. Parsley and other hosts. But that kind of a detailed knowledge and study won't be often available or the resources for it. So presumably you have envision a tiered management strategy where your management recommendations parallel the extent of your understanding of biology of pathogens.
Absolutely. Yeah. So for example, often before I would even begin a research project, what I ask growers is, what are your options? How are you going to manage this? What are the things you are likely to do? And once I get those answers, then I can actually target the study. If they know it's if they know it's bacterial and they only have 2 options for control, that's all we need to know. Is it bacterial? Is it not? Right? And so it really depends what options are available to the growers. So with the mushroom growers, they're very innovative. They have some extra money. So they would be willing to invest in products. They would be willing to invest in development of products. And so looking at, for example, these development of a biocontrol for them makes sense, okay? Because that high value cash crop would make sense. That wouldn't necessarily make sense for lettuce, okay? And so my first question is always to the producers: what kinds of things do you have money for? What kinds of things are you willing to do? Does that make sense? Does that answer your question? Okay.
Carolee, we don't have many mushrooms produced in New York. What sort of options do Pennsylvania mushroom growers have for control or reduction of bacterial diseases of mushrooms?
It's such a fun crop. Oh my goodness. When I moved to Pennsylvania, I thought, how lucky could a Pseudomonas girl be, right? That you've got all these, and actually Pseudomonas is involved in the stimulation of mushroom growth. So the actual fruiting body formation by the fungus is stimulated by Pseudomonas putida-like organisms. So that's a whole nother ball of wax, right? You don't want to kill off the good guys, and you want to kill off the bad guys. And so, so one of the things that conventional growers can use is chlorine. Organic growers cannot use chlorine, okay, for example. The primary methods for control are sanitation, steam, so they steam their whole rooms. They can steam the entire room, and humidity control. But that isn't optimal for, like the things that are optimal for pathogen control are not optimal for growth. And so they're constantly doing this fine balance. And in the organic systems, they really tip toward disease often. And so, in fact, we were just awarded $1 million ORI grant. So that's the Organic Research Extension Initiative funding to work on this and a couple of other diseases in relationship to biological control in particular, and microbiome analysis more generally. So they really have humidity control, sanitation, and chlorine. And then if you take away chlorine for the organic folks, it's sanitation and humidity control. And so they're really looking for other options. And I think, you know, they're already starting to get a picture that their disease problems are more complicated than they once thought, right? So that really is my job, is to let people know that their disease problems are more complicated than they thought. And they can't just deal with one breeder. They need to work with us too.
Carolee, I'm curious of how all this flux in taxonomy, how that is intersected with international trade barriers and regulatory sort of things. Are they keeping up with the changes, or are they? Yeah,
not at all. Yeah, in fact, I have been working with APHIS in particular, and just the permitting section to try to get them up to date in the understanding of what the bacterial taxonomy is, because some of them really do understand it. And as they bring new people in, you know, we want to make sure that they're up to date as well. So I would have to look at my calendar, but I believe there's a visit to APHIS on my calendar soon to give the whole 2 or 3 hour version of bacterial taxonomy at a glance. The folks from the Penn State Department of Agriculture asked me to come and talk about bacterial taxonomy, and I said to them, well, I feel like I've done an adequate job when I've talked for a week. How long can I have, right? And so, I know, a week of bacterial taxonomy. I always try to make it fun. So, I've had a number of people come visit me for a week so that they can do it. Other questions? So now you know what translational taxonomy is. Perhaps.
Well, if there are no further questions, let's thank Carolee again.
This has been a production of Cornell University, on the web at cornell.edu.